Okay, welcome back. Again, we are in the second part of the course. This is Applications of Biotechnology. From the course, Issues in Biotechnology, the Way We Work with Life. We're in the medical biotechnology section, and we are considering applications in tough times. That is, we just got done looking at pandemic flu, and we will consider now biotechnology as it applies to bioweapons. So bioterrorism, I will first present a history and an update, and then look at bioweapons now as they exist as an emerging threat and the possibilities for the future. But first, since the anthrax attacks of 2001, which many of you may remember, if a large-scale bioweapons attack were to occur in New York, Boston, or Providence, do you feel as though you are informed for proper evacuation, or response procedures? You could rate this A, well-informed, B, somewhat informed, C, unchanged, D, confused, F, totally unprepared. And maybe that is how we could rate Homeland Security according to your response. But since the anthrax attacks of 2001, where you may remember the media frenzy that occurred about that, and rightfully so, do you feel that the threat of a major bioweapons attack as addressed by Homeland Security has A, been eliminated, B, greatly reduced, C is unchanged, D has worsened, or F has significantly increased? I'll just let you respond in that poll. But also, since September 11th, I feel that our threat from terrorist attacks has been A, B, C, D, or F. Since 9/11, I would grade the U.S. government on addressing Homeland Security, A, B, C, D, or F. We've recently seen a lot of activity globally in suppression of terrorism. But bioterrorism is of a different sort. Bioterrorism is the deliberate use of microbial agents and toxins, specifically as weapons. We can see that the U.S., if not the global community, is vulnerable due to international political and economic instabilities, state-supported and tolerated terrorism in some parts of the world, and a freedom of access of knowledge, as we just discussed in the previous lecture. This results in a global vulnerability due to increased technological capabilities, such as the ability to culture and study these organisms, as well as synthetic biological approaches that will remain and exist into the future. So effective response requires coordination then of medical public health systems, as well as emergency management and law enforcement should a bioweapons attack occur, whether that's homegrown or from without. So a brief history. We could see that in Exodus chapter 9, verse 3, as the notation of the fifth plague visited upon the Pharaoh of murrain carcasses and pestilence. Murrain refers to literally dead animals. How those animals came to be that way is up for speculation, but given that it was the fifth plague, I reckon that was not a great scenario. The 14th centuries, the Tartars catapulted plague-infected corpses into the city of Kaffa. And in the 18th century, the invading British into North America used blankets to transmit smallpox to Native Americans. And so there was a wave of smallpox infection that preceded the takeover of North America by the Europeans. In World War I, Germany used glanders to infect livestock bound for Allied forces. And in 1932, Japan conducted large-scale human bioweapons field trials, sadly in Manchuria and in China. From 1942 to 1973, as I'll describe in a moment, the US had a program of offensive and defensive bioweapons, including human volunteer exposures and field trials on weapons or innocuous agents which would behave as weapons if they were deployed. During the Cold War, both major superpowers had active research and production capabilities in many of the offensive weapons that I'll describe in the lecture tonight. In 1972, the Biological Weapons and Toxin Convention formally banned the use or development of BWs. And in 1979, 68 deaths occurred in the former Soviet Union due to inadvertent release of weaponized anthrax from a military facility there that eventually was closed after the fall of the Soviet Union and may have been raided by outside forces. In the United States, one really historic but rather odd event occurred in the Dalles, Oregon, where a cult, the Rajneeshi cult, deliberately contaminated commercial salad bars with salmonella in order to try to tilt election results in Wasco County, Oregon. This group had already commandeered the town of Antelope and was looking to take over the Dalles, but in order to try to sway that election, contaminated salad bars with salmonella in order to prevent voters from reaching the polls. In the Persian Gulf War, that is the first one, Iraq was known to weaponize biological weapons agents. And I'll talk about that because there is the existing controversy about weapons of mass destruction and their supposed involvement in the Iraq war after 2001. In 1995, sarin nerve gas attacks occurred in Tokyo and killed 12. 1998 to 1999, there were 50 anthrax hoax attacks that weren't actually anthrax spores themselves, but powdered white powder that was perpetrated as an anthrax hoax, which more or less set the stage, if you will, for the deadly anthrax attacks that followed on the heels of 9/11. And if you recall, post 9/11, the frenzy that existed after the suicide attacks on New York and Washington and the plane that went down in Pennsylvania, certainly many people thought that the anthrax attacks were linked to those events. So what about bioweapons in Iraq? Let's talk about that first. What is the history of bioweapons development in Iraq that preceded the first Persian Gulf War, and then the Second War. What is known actually about bioweapons development in Iraq? And then a brief history on bioweapons in the United States. So what was the situation for weapons of mass destruction in Iraq? The Al-Hakim single-cell protein plant was the major facility for the production of biological warfare agents in Iraq prior to 1980. And it was under the watchful eye of the United Nations Special Commission, and it was destroyed by Iraqi workers in May and June of 1996. But let's give a chronology briefly of some of the events that led up to that. According to the UN Special Commission, Iraq's biological weapons program ran roughly from 1973 to at least 1991 and possibly after that. Iraq claimed that the program began with the establishment of the Al-Hazen Institute as a dedicated bioweapons facility. And they studied in that facility from 1974 to 1978 botulism toxin, anthrax spores, Shigella, cholera, as well as some viruses, camel virus. The institute was closed in January of 1979 because of fraud committed by its senior staff. But in 1987, Iraq transferred the bulk of its biological weapons work to the Al Salman Technical Research Center in order to maintain the program's secrecy, and pushed work on anthrax spores and botulism toxin. And then viruses and genetic engineering were introduced in 1990. In 1988, research was conducted on gas gangrene and in mycotoxins. So you can already see, just by this review, that there was a range of bioweapons research for offensive purposes that was documentable by the UN in Iraq prior to the 2000 invasions. Iraq admitted that it had weaponized biological agents between 1990 and 1991. And these were being developed for missile warheads, aerial bombs, aircraft drop tanks, pilotless drone-like aircraft, and helicopter spraying. I put together a list or a summary of some of the bioweapons that had been focused on in Iraq, including aflatoxin, which Iraqi scientists studied on how to produce liver cancer. Certainly a long-range effect, but also anthrax spores, which when weaponized, as we saw in this country, 1 gram of dried anthrax spores goes a long way. And it's been estimated that the Iraqi constituents could contain up to 10 million lethal doses per gram. Botulism toxin is the most poisonous substance known. The average man would only have to inhale about 70 billionths of a gram of that in order to be fatal. Cholera was worked on. It's passed on naturally versus contaminated food and water, and as a weapon would be most likely to be used to poison water supplies. Clostridium perfringens causes gas gangrene, which in turn causes toxic gases to form in body tissues. Mycotoxins are a family of poisonous compounds made from a mold that grows on wheat, millet, or barley, and I'll get into the possible use of agricultural bioweapons and how those might be transmitted in a while. Iraq worked also on camel pox, which rarely infects humans, but largely as a model system to learn more about how using smallpox might be used as a biological weapons. Hemorrhagic conjunctivitis, rotavirus that causes diarrhea, and smallpox, which could be inhaled or absorbed by the skin, were all being worked on by Iraq bioweapons facilities. So, as you know, in retrospect, Iraq also had chemical weapons which it deployed on some of its own people in the Kurdish population in the 1980s, killing large numbers. And by 1991, the United Nations had established its special commission in order to keep track of chemical agents being used by Iraq. So there really is no question in this case whether or not Iraq had biological weapons that were being researched for deployment as weaponized agents, and it had in fact used chemical weapons on its own people. The nuclear weapons were suspicious, are suspicious, and really, I think as we know now, largely unfounded. So what about bioweapons in the United States? That's how a axis of evil country pursued bioweapons. What actually happened in the United States? The United States began an offensive biological program in 1942 under the direction of a civilian agency, the War Reserve Service, the WRS. And the program was significantly expanded during the Korean War from 1950 to 1953 at a new production facility that incorporated adequate biosafety measures was constructed at Pine Bluff, Arkansas. And a number of volunteer studies were performed in a large facility, a large sphere that was built at Fort Detrick that became known as the 8-Ball to test weapon systems largely for disseminating various pathogens. So the United States began research on bioweapons as early as 1942 and had an active program until President Richard Nixon closed the American Offensive Biological Warfare Program. But in 1969, the budget for chemical and biological warfare research was reported to be about $300 million, with about $5 million devoted to herbicides, such as I mentioned in a previous lecture, such as Agent Orange, designed to kill food crops or strip trees of foliage to deprive enemy forces of ground cover. So that would be another type of biological warfare that could be exerted on an existing country to kill crops, whether those crops be opium in Afghanistan or wheat in a country used to feed its supporting military or its civilian sources. But also I would want to point out that there were a number of cities that were unwittingly used as field study laboratories, if you will, to test the aerosolization and dispersal methods of such agents as Aspergillus, Bacillus subtilis, and Serratia marcansicans, used as more or less to simulate what would happen in experiments that were conducted in New York City subways, San Francisco Bay, and in other sites around the United States. Also, there were 456 cases of occupational infections that were acquired at Fort Detrick during the offensive biological program there. And those occurred at a rate of less than 10 infections per million hours worked. We could note that that's a very high level of safety, 10 infections per million hours worked. We could also note that with 456 of them, that there were a lot of million hours worked in that facility. I'll let you look more closely at this slide, but it shows that in some of the tests that were done in these field studies by the United States under the auspices of biological laboratories studying the dispersion of possible simulating biological weapons effects, that in one such test, travelers to the Washington National Airport were subjected to a harmless bacterium where traps then were placed throughout the airport to capture the bacterium as it flowed through the air, and that laboratory personnel dressed as travelers carrying briefcases walked the corridors and without detection sprayed the bacterium into the atmosphere in order to conduct that test. In New York, in a subway, a light bulb was filled with the same harmless bacterium and dropped onto the tracks, and that organism spread throughout the system in 20 minutes. So we've known about these capabilities for a long time. So you could see then how a weaponized bacteria such as anthrax spores, we already know how it would travel. And traps were set up to monitor. There's predicted that a dangerous organism could have killed thousands of people, and this would still be the case today. In San Francisco, a U.S. Navy ship that was equipped with spraying devices operated by Fort Detrick personnel sprayed a innocuous bacterium. It's a non-pathogenic microorganism that's easily detected while the ship sailed through San Francisco Bay, and it was shown to have spread 30 miles by monitoring stations. So a ship today dispersing on a windy day in San Francisco could disperse an agent 30 miles in a radius downwind. Let's keep this in mind when we look at some of the popular bioweapons that could be dispersed that are available now. This is a picture of the front of the building of the United States Army Medical Research Institute for Infectious Disease on the grounds of Fort Detrick. Fact or fiction? It's often brought up to me that Plum Island off the coast of Connecticut, a short ride from Orient Point, Long Island, or from the town of Lyme, could have conducted biowarfare research. And it's been speculated amongst the population in Connecticut that this may have been the origin of Lyme disease, and that if the United States was conducting bioweapons research out at Plum Island, that's a rather short swim for a deer that could have communicated that disease back to the community of Lyme. But actually, Lyme disease is known to have occurred or suspected to have occurred prior to 1975, likely originating in Europe before it migrated to the United States. But what about the possible threat of bioweapons to agriculture? I had mentioned mycotoxins, aflatoxins, or the ability to utilize herbicides to take out existing crops. Certainly those are all viable opportunities. The dispersal of weeds, as strange as that might sound, but to get even more elaborate, you can imagine what would happen to the beef industry if someone were to sneak into a cattle production facility with a syringe filled with mad cow, relatively undetectable. I don't want to give anybody ideas here, but these are actual real possibilities that possibly we should all be aware of. What about the use of agricultural weapons as an offensive weapon? Well, that's already been done considering Agent Orange as a defoliating agent, but what about the ability to spread a disease to a country's crops that country is dependent upon for its survival? In the past, the United States has applied embargoes of agricultural crops to control policies. The wheat embargo to the Soviet Union in the 1980s had a significant policy impact during certain negotiations. And remember, I brought up the possible influence that the United States possibly could have levied on its control of global agriculture. Is it, and you might argue ethically, is it better to drop bombs or is it better to withhold food? I don't know, but that's certainly a situation that could have been approached, or possibly still could be. So again, the notorious attack by cult members that contaminated salad bars is a well-known, homegrown terrorist attack that used a bioweapon in Oregon using salmonella in an effort to tilt an election. And you can see that people got the answer correct here. What about the anthrax attacks of 2000? They began on Tuesday, September 18th, 2001, about a week after the September 11th attacks, and therefore many people considered that they were linked. Letters containing anthrax spores were mailed to several news media offices and 2 Democratic U.S. senators. Those attacks killed 5 people and infected 17 others and caused the contamination of those buildings. According to the FBI, the ensuing investigation became one of the largest and most complex in the history of U.S. law enforcement. A major focus in the early years of the investigation was in fact a bioweapons expert named Stephen Hatfield. Stephen Hatfield was eventually exonerated, but it probably ruined his life. It probably ruined his career. Another suspect, Bruce Edwards Ivins, became a focus of investigation around April 4th, 2005. Ivins was a scientist who worked in the government's biodefense lab at Fort Detrick in Frederick, Maryland. And on February 11th, 2007, Ivins was put under periodic surveillance, and an FBI document stated that Bruce Edwards Ivins is an extremely sensitive suspect in the 2001 anthrax attacks. On July 27th, 2008, Bruce Ivins killed himself with an overdose of Tylenol. How scared should we be? Well, again, this was a homegrown terrorist. And in fact, the United States has seen several effective homegrown terrorists since the Oklahoma City bombings, the anthrax attacks, the DC snipers, etc. Could you suit up for safety and everybody walking around in their own BSL 4 level-like facility? No, but certainly to have bioweapons experts that could mitigate an attack should it occur in an airport, a stadium, or some large facility is certainly part of what Homeland Security should be all about these days. What does the homegrown terrorist look like? Here's a photograph of Bruce Edwards Ivins. I don't know what a terrorist should look like, but he looks like, nearly anybody. He was educated, PhD from the University of Cincinnati. His religion, Roman Catholic. Worked at the Frederick Memorial Hospital and died in July 29th, 2008 at the age of 62. This was the cover of the New York Post during the anthrax attacks. Stricken New York postal girls sends that message to the terrorists. But, prior to 9/11, when people talked about bioterrorism, it was pretty low on the list of consideration for threats to Homeland Security. In fact, the word Homeland Security hadn't been invented yet, but considering threats to the shores of the United States, bioweapons was not really very high on the list because most of these bioweapons would rely on transmission. And how would you adequately disperse a biological weapon? Well, smallpox, for example, is person-to-person transmissible. So I think, before suicide delivery agents came to our consciousness abruptly on 9/11 on 2001, we really didn't consider the likelihood that someone would infect themselves with a disease and then walk onto a crowded airplane or into a crowded stadium. Why? Would someone get into a plane and purposefully drive it into a skyscraper? Well, now we have to deal in our consciousness with the possibility of suicide delivery agents and the ability that people would sacrifice their own lives to jeopardize or eliminate others. So the ideal offense agent now would either be a suicide delivery agent or one by aerosol delivery that's easy to disseminate, stable over wide temperature range, and have a long shelf life. It should be inexpensive, able to be produced in large quantities, and reliably cause high mortality, morbidity, or at least widespread social disruption. The CDC has prioritized biological weapons then according to categories A, high mortality, major impact, B, moderate morbidity, and C, emerging pathogens. And again, true to this course, we can put on our futuristic goggles here and look a little forward-reaching. So in category A, we have anthrax caused by Bacillus anthracnus, botulism caused by Clostridium, the plague, Yersinia pestis, the pox virus, or also known as smallpox, variola major and variola minor, tularemia, the Francelia tularensis, and various viral hemorrhagic fevers. These collectively known as VHFs are all RNA viruses shown of the different families listed here. In category B, these cause moderate morbidity. We have Q fever, Coxella, brucellosis, the Brucella abortus, glanders, Burkholderia, Mallia, and viral encephala, and ricin toxin, which is caused by castor bean extracts, and Staphylococcus enterotoxin B. And here's a list of some other Category B infection agents and Category C that might be emerging threats, such as hantavirus, tick-borne viruses, yellow fever, and tuberculosis that would be drug resistant. The reason that I mentioned that study that was conducted in the San Francisco Bay Area is to give you an idea about the casualties that could occur from a hypothetical aerosol attack. If the agent were Rift Valley fever and the downward reach were, say, 1 kilometer, the number of dead might be 400 in a populated area, and about 35,000 that would be incapacitated. You look at something like brucellosis, could have a downwind reach of about 10 kilometers and could incapacitate about 125,000 people. A tularemia or an anthrax exposure could result in 95,000 dead and upwards to be over 125,000 incapacitated. So a bioweapons aerosol attack of anthrax, say someone throwing a suitcase off the top of the Empire State Building, or that ship driving into San Francisco Bay, could have such an effect with anthrax spores that are already known conventionally to be able to be weaponized by such a guy like Bruce Ivins. Detection is likely to occur before circumstances, the illness is likely to occur before circumstances are known, so that people would start to be affected, but we wouldn't know exactly that it was from a bioweapons attack, and that the characteristic symptom complex would be suggestive of a bioweapons agent, but first responders might not be familiar with, say, something like smallpox or anthrax. Like how common is that to a first responder in the Northeast United States due to the occurrence of relatively rare infections? Temporary clustering would be the first clue, perhaps, but this might take days or weeks. And a concurrent disease in humans and animal populations in that region, especially for these diseases that are known to infect both animals and humans. So in a bioweapons attack, the unique challenges will be first response because of A, B, C, or D shown here. So the problems would be that civilian targets would be unaware of a release. Military targets would be more predictable. In fact, in the Gulf Wars, military personnel were vaccinated in some cases against possible biological weapons and had equipment that they carried into the military theater to prepare them in case of bioweapons or chemical attacks. Casualties would be staggered in their presentation and might be geographically dispersed. We can imagine a bioweapons attack happening simultaneously in several US cities, say LA, Chicago, Boston, and that these simultaneous attacks, whether transmissible or not, would be difficult to coordinate. Physicians clinically inexperienced due to the rarity of these would further delay a response by first responders or military personnel. So the release of a biological agent then could fly under the radar, so to speak, for quite a while before it was discernible, and the casualties would not be immediately present and would escape then medical attention. And depending on the agent, firefighters, police, and paramedics may not be involved initially, but physicians would be most likely the ones to be involved. Now, if you compare this to other weapons of mass destruction, you can get a sort of an idea of the scope and how this would differ from other weapons of mass destruction. In the case of a nuclear attack, the area involved is large. We know that. Detection, well, that's pretty easy. Clinical incubation would vary with dose and proximity to the blast. And the medical intervention is fairly limited. In a chemical warfare response, the area involved would be rather limited. Remember the sarin gas attacks in Tokyo or the Kurdish attacks. Limited somewhat, but could be upwards of thousands of deaths. Detection would be moderate, depending on the agent. Clinical incubation is minutes, if not seconds, and medical response is somewhat limited depending on the time that's allotted. In a biological weapons attack, the area involved would be moderate to large, detection difficult at best, incubation could be days to weeks or in some cases months, or some agricultural applications, years. Think mad cow disease. And clinical intervention, effective with some and non-existent for others. Well, I already mentioned the so-called Sentinel-6 of Category A virus or biological agents. These are so-called naturally occurring possibilities. So let's go through them. Anthrax, smallpox, plague, tularemia, botulism, and hemorrhagic fevers. Anthrax, we already saw, could have an impact. It's relatively simple to grow in the laboratory, actually. Anybody that is skilled in the art could produce Bacillus anthracnus. Where would you get it? Well, a dead cow could have this naturally occurring in Texas. If you're a veterinarian, you're pretty much well schooled in the presentation of anthrax as it occurs in large animals, and attaining a sample is not too difficult. Culturing it in the lab is published and pretty well known. Weaponizing it maybe a finer art. Anthrax epidemiology is infected animals or contaminated animal products can carry this, and most infection would come from skin contact to an animal. Veterinarians can get this by touching dead animals. People can also get it from eating raw or rare meat or inhaling the spores from soil, contaminated soil around a dead animal. And there's usually no person-to-person transmission. Skin infection usually occurs directly through a wound site. Lung infection can occur from inhaling the spores. So contact on the skin and its entry is usually due to minor cuts and abrasion. And the symptoms then in that case would be a rash, whereas for inhalation anthrax, inhalation will cause flu-like symptoms with a fever, chest pains, could lead to breathing problems, shock, coma, and then eventually death. It can also be ingested through the GI tract. Spores can enter the digestive tract through undercooked or raw meats from contaminated animals. And this would include nausea, vomiting, and perhaps bloody diarrhea, which could be treatable. So from inhalation infection, we would consider that the most serious possibility for a bioweapons attack, and it starts with flu-like symptoms 1 day to weeks later, which could result in severe breathing problems and shock. Cutaneous anthrax presents as shown in this slide here, largely contacted directly through a small lesion or abrasion on the skin. So the greatest risk is the aerosol delivery for infection, which could occur within 24 hours of release. This could occur, as we saw, through mailed envelopes containing powderized spores. The spores typically of anthrax would clump together if you grew these in a laboratory. So the development of specialized strains that the spores would not stick by either clumping together due to, say, moisture or static electricity is a part of culturing strains specifically for bioweapons. So this is merely really in any of these laboratories, whether Iraq or Fort Detrick, a matter of experimentation, hit or miss kind of thing. We saw that the 9/11 perpetrators trained on biplanes. Certainly, we don't know if that was part of their, just their training to fly a plane, but the delivery of aerosol methods from small biplanes is certainly a possibility for weaponized anthrax. I mean, given the aerosol delivery agent field trials that I discussed about San Francisco Bay, a biplane flying over Manhattan could have pretty serious implications. Inhalation anthrax as a biological weapon is certainly a possibility. If there is any good news, it's not contagious, so that only those exposed to spores get sick. There's no unintended victims, so to speak. That spores, however, do last a long time. So we saw that in the attacks on the US senators, that all of the air conditioning ducts had to be removed and cleaned, and that early symptoms are not specific, but this is a very effective agent, 85% fatal as an aerosol. It's also easy and cheap to produce. So if you consider making an atom bomb, that's actually pretty easy to do. It's a lot harder to come up with the materials. In terms of a bioweapons attack using anthrax, pretty easy to come by, very cheap. Facilities could be maintained in the back of a semi-trailer that could be rolled around. Treatment of anthrax, doxycycline works. So it's the drug of choice, and Cipro was selling off the shelf in 2001. So again, you'd almost have to think about who would get this and when and how if there was a large-scale bioweapons attack. And you remember, some of you, during that 2001 attack, that no one knew exactly where the next event was going to occur. Florida, New York, Connecticut, where was the next letter going to be opened? Could it have been your own mailbox? Everyone started hoarding Cipro. Penicillin is the cheapest alternative if cultures prove to be sensitive, but developing penicillin-resistant strains could be part and parcel of a bioweapons approach. And that people were not only stockpiling Cipro and doxycycline, but taking it prophylactically. So these kinds of drugs literally were selling off the shelves due to hysteria surrounding the early bioweapons attacks. Well, I'll get into this in the later part of this lecture, but genetic engineering of bioweapons, let's face it, antibiotic resistance genes can be purchased or traded. You can get these cassettes, as I discussed earlier, for transgenes, promoter, coding region, termination signal in a plasmid that, say, would encode resistance to aminoglycosides, kanamycin, neomycin, hygromycin. These are readily available. Penicillin resistance, Cipro resistance, these can be generated. How difficult is an experiment is it to make a anthrax that is Cipro resistant? While you're at it, why not just make it penicillin resistant? This kind of strategy could be applied to nearly any bioweapon. So you think you have a very common, naturally occurring bioweapon. Here, just take Cipro. Well, maybe not so. What if you were to end-to-end tail a lot of resistance factors as part of a bioweapon strategy? So standard precautions for cases, but also suspicion of even one case should prompt notification of state health department and local law enforcement agencies. It's not common for a person in the northeast of the United States to contract anthrax. If you get a letter with any white powder in it, we would know these days to immediately contact authorities. There have been numerous hoaxes perpetrated since then. Those people are treated, that's a felony, and it's treated as a security issue and punishable by, I think it's 10 years in prison. So if anybody is thinking of a hoax, this is anything but harmless and seriously punishable. Let's move on. Plague. Plague is transmitted as Yersinia pestis, and well known from the Middle Ages, the plague. The right stain identifies Yersinia pestis in a blood smear of a patient as this bipolar safety pin staining of the bacteria. So even though it went through Northern Europe, consider it as a biological weapon, outbreak would pose a serious threat. It could create widespread panic because of its known reputation and devastation. And like anthrax, an attack of aerosol Yersinia pestis would cause highly lethal pneumonia. The attack rate would be dependent on the agent used. So pneumonic plague would be highly contagious and spread by sneezing or bites from an infected flea. So a suicide delivery agent of plague could enter a crowded subway, a Super Bowl, arena, you name it. And it's almost 100% fatal if not treated within 24 hours of onset. The plague in the Middle Ages was largely communicated by fleas that bit infected rats. The incubation period is 1 to 6 days, typically brief. A skin lesion appears and necrotic digits in the advanced part of the disease. A primary bubo, or a primary bubonic plague symptom, is shown in this slide here. And this is in its more advanced stage. Notice the presentation on extremities and the digits. The detection, it would be unable to pick up an aerosol plague cloud, and no readily available widespread rapid testing capabilities currently exist. Clinically aggressive, and it would be little time for diagnosis. So Given plague as a biological weapon, therapy recommendations would also be doxycycline or Cipro, but again, I would counter this by the capabilities of making these weaponized agents resistant to Cipro or doxycycline. So it's contagious only until 48 hours into antimicrobials. Moving on to smallpox as a bioweapons agent. Variola major and variola minor have been around for a very long time, and the stability in aerosol form is inversely related to temperature and humidity. Its infective low dose of about 10 to 100 organisms makes it a weapon of choice. It's also efficiently spread person to person. This is a real bummer. Secondary attack rates 25 to 40% amplified in essentially all children, and most adults currently are non-immune. Only limited vaccine supplies are currently available. This disease was almost extinct on the planet, but now we are open for current infections. Smallpox as a biological weapon would be very effective as a suicide delivery agent, and it would take a fair bit of time that an infected person would have in order to communicate this person this disease from person-to-person contact, say in a large city. So several suicide delivery agents could easily get onto a plane infected from another country, not wearing any sort of other explosive device, get onto that plane, infect the people on the plane, who would then subsequently infect other people as they left. Those people could then infect the suicide delivery agent then could go on into large urban areas or attend a large sporting event or other populated areas and have quite an impact. So person-to-person spread for any of these biological agents is a large threat, especially with the incubation period of 7 to 17 days. It's 10 to 12 days before a rash appears. The clinical presentation certainly isn't pretty. Fatal smallpox has in history ravaged large populations. In its clinical presentation, a rash scabs over in the first 10 to 14 days after onset, and it may be attenuated or atypical in variola minor or partially immune individuals. Hemorrhagic or malignant varieties are frequently fatal. The gentleman depicted down in the lower right of this slide has a particularly malignant variety of variola major that was hemorrhagic. It's communicable from onset of rash until separation of all scabs, roughly about 3 weeks. It's most contagious during week 1 due to high viral titer in saliva. So clinical diagnosis would require an index of suspicion. So how would this come about in the first few days of a bioweapons attack that was coordinated around several cities in the United States? The good news is vaccine is highly effective. Large-scale preventative programs are currently not feasible, but could be instigated and implemented if such a weapons attack were imminent. So isolated suspected cases at home would be possible in order to contain an attack if such a thing happened. You would imagine then that a strategy, if suicide agents were deployed to the United States, that they would be staggered. You could imagine one wave coming all at once, and then a second wave and a third wave, and so on. And again, we wouldn't have thought of this prior to, say, people wearing suicide vests and so on. Botulism. Botulism is relatively unlikely, but we saw that Iraq had an active program in biological weapons for the use of Clostridium. It can be weaponized. It has been weaponized by several countries, including the Soviet Union. It was studied by the US, and it was aerosoled by Iraq. It would kill about 10% of the population that was exposed if adequately dispersed. It could also be used to contaminate the food supply. We're well aware of food poisoning by botulism and recalled food safety project products that were not bioweapons. The early symptoms include drooping eyelids, weakening jaw clench and trouble swallowing, blurred vision and trouble breathing. Eventually, nausea and vomiting would be a sign that the germ had been eaten. And basically, individuals would look like this and might also present with dilated pupils. It's not contagious, but mortality rates, if exposed to a high enough level, are sufficient. Botulism vaccine is available. Tularemia is also a Category A biological weapon, weaponized by both the United States and the former Soviet Union. Small animal reservoirs are possible, better known as rabbit fever, and transmission by aerosol or direct contact with animal tissue is possible. It can live for weeks in soil, water, and dead carcasses. It's highly infectious. Its incubation period 3 to 5 days, and fatality rates are about 30% without treatment. Begins with a flu-like symptom, and over the next few days, painful breathing and cough begins. Clinical presentation of tularemia might look something like this. Its treatment is also doxycycline or Cipro and respiratory support. There is no person-to-person transmission, and there's limited availability of a vaccine. Lastly, but not least, perhaps most graphically, hemorrhagic fevers, such as Ebola viruses, which upon infection would cause massive and profuse bleeding virtually out of every orifice until death. As a bioweapon, it's considered extremely unlikely, both because of its difficulty of handling and because of its transmission would have to be by person-to-person, blood-to-blood contact. So a terrorist would have to deliberately infect himself, travel to a major city to spread the disease by sexual contact or by sharing needles, or perhaps blowing up in public. So most people infected with Ebola die within 1 week with a lot of suffering. So in the case of the latter attack, it would be graphic. And isn't that the goal of many terrorist attacks after all? Sudden fever, headache, muscle pain, nausea, vomiting, chest, cough with chest pain, red rash, and then profuse bleeding leading to death. No known cure. What about bioterrorism and counter-defense? What goals might an attack have on the agricultural sector? Well, attack of food supply of an enemy could occur. This has occurred in the past, and the altering of supply and demand patterns on a commodity might vastly affect an economy. We know that one mad cow in California could offset cattle industry tremendously in terms of not only its exports, but locally, let alone if that were undetected and the meat resulted in infecting domestic humans, as we saw in the UK several years ago. That now locally is controlled mostly by USDA inspection services, and I think they have been very good at detecting mad cow disease, so lessening the likelihood of an agricultural sector attack on that. However, it's also considered less likely that a terrorist would, say, spray a crop with an infectious disease that might diminish a crop like maize or soybean in the US, because it's just not as spectacular as running a plane into the World Trade Center. How will genomics and DNA-based technologies change the biological warfare threat? Again, I invoke you to put on your futuristic goggles. We can make viruses. We can make antibiotic resistance genes and incorporate those into existing bioweapons or ones that now might be more creatively designed. High-tech agent design is an option available only right now to sophisticated players. Is that really true? Like I said, some countries are putting a lot into development of a nuclear warhead. It's a lot cheaper and requires a lot less technical sophistication in order to develop widespread biological weapons. The appropriate course of action for a biological attack would be to contain, confirm, clean up, direct appropriate treatment as needed. And as I think I detailed in the beginning about these questions about how prepared we are post 9/11, that's been 11 years, 12 years on. How prepared are we for a current biological weapons attack by any of the agents that I now have just described? The objective of a terrorist, after all, is to destroy normal day-to-day life. If a bioweapons attack occurred in this country, fear would be almost the largest enemy. As we saw with the anthrax attacks, who knew who would get the next letter? So you can look up on the internet now how to make a biological weapon. So I printed this one out. Let's make a biological weapon. Now, given that you've been in this course so far, I'll just pose this as a little homework assignment. Can you pick out the flaws in this guy's argument? On the other hand, you can think about how relatively unsophisticated a technology it is in order to weaponize some readily available biological agents. As I said, you can also find out how to make a nuclear bomb. In fact, it's relatively easy. I described one to my 6th grade teacher for show and tell once, drawing it in a diagram about how you can force 2 pits of refined plutonium against each other to cause a nuclear fission. I mean, if a 6th grader can do it, that's just the diagram, however. Coming up with the raw materials there is under the watchful eye of a lot of people, gratefully. However, to make a bioweapons agent is, as I said, a lot less sophisticated, and the agents are cheap and readily available. For bacteria, start with a known agent. Why not? genetically engineer it with antibiotic resistance, weaponize it as needed, engineer antibiotic sensitivity for self-protection. Viral, start with a known agent. You might want to genome sequence it. Use of synthetic biology or creative components. Agricultural, start with a known agent. So these things are well known to the public, and I suppose that, like we were saying before, the dual use of knowledge component applies to these areas, certainly. And now synthetic biology moving forward. Do we really take the threat of biological weapons as seriously as perhaps we should? Is synthetic biology a larger threat to world peace than nuclear war? Managing nuclear war has been one of balance and control. We have more bombs than you, and therefore you better not. Hasn't been that the way it has worked. What kind of a strategy do we need to politically invoke in order to stop a threat of future bioweapons as a result of creative synthetic biology? Who's considering this? Have you all heard of this? Really? We hear every day about the threat of nuclear war from Iran or other so-called rogue countries or countries that have them now. What about pandemic flu as a bioweapon? We considered that possibility in the last lecture. We saw that the 1918 flu was resurrected. What if somebody wanted to just spread that around? Person-to-person contact. The sequence has been published. Dual-use dilemma. Could the world stop it if it could? Is it only a matter of time? Well, research such as the recent publication of the mutability of H5N1 to make it more transmissible certainly raises the bar of concern and spurs on the controversy about publication and dual use. An engineered avian flu could kill half the world's population. So there is an obligation to prevent the next dual use controversy, as I pointed out in the previous lecture. I side with the force that favors knowledge, and that certainly ignorance is bliss. Controversy spurred a moratorium, but it certainly wouldn't spur access to this knowledge. Or how unique is Ron Fortier? Don't you think really anybody could reiterate that research? Not anybody. I mean, he's a talented researcher. There's no question about that. But given the desire to pursue something like that, so as pointed out in this movie by people in the military, suspecting that the agent that is depicted in Contagion could have been a bioweapon. The CDC researcher comments that maybe the real terrorist is a chicken in Southeast Asia. I think appropriately, because probably evolution of these viruses is occurring faster than human innovation could do it anyway. I don't mean to diminish any concerns about the ability to manipulate bioweapons. And again, that brings me to Orcs and Crake, and that some say the world will end in fire, and some say in ice. From what I've tasted of desire, I hold with those who favor fire. But if it had to perish twice, I think I know enough of hate to say that for destruction ice is also great and would suffice. Again, Robert Frost.